Good afternoon, friends. Today we will discuss about radioimmunoassay. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. Radioimmunoassay, also known as RIA, is a very sensitive in vitro assay technique. It is used to measure the concentration of an antigen by use of specific antibody. The antigen can be a hormone, an enzyme, a drug, a virus, or any other antigenic substance against whom we have specific antibody. RIA was developed by Solomon Besson and Rosalind Sosmaniello in 1959. They used radio-level insulin. They used radio-level insulin to estimate serum insulin. Since the initial description of yellow assay, many technical variations have been developed to make the method more rapid and reliable. Radioimmunoassay is a competitive immunoassay in which there is a competition between radio-labeled antigen and unlabeled antigen for binding with a limited amount of specific antibody. It involves the formation of antigen-antibody complex. In order to perform a radioimmunoassay, we require number 1, a sample containing the antigen of interest. Number 2, complementary antibody and number 3, radio-labeled version of the antigen. Radio-labeled version of the antigen. A known quantity of antigen is made radioactive frequently by labeling with iodine 125 or tritium, which is attached to the exposed tyrosine residue of proteins. The antigen present in the sample is known as unlabeled antigen, which is also known as cold antigen. In this technique, a sample containing the unlabeled antigen and the complementary antibody are incubated together. As a result, there will be binding between these two and antigen-antibody complex will be formed. This is followed by the addition of radio-labeled antigen. Now the radio-labeled antigen will compete with unlabeled antigen of the sample for binding with antibody. As a result of this competition, some of the sample antigens will be displaced from antibody binding site and in their place, radio-labeled antigens will bind to antibody. The more the sample antigen present, the less the radio-labeled antigen is able to bind to antibody. This figure illustrates the principles of radioimmunoassay. As already mentioned, in this method, there is a competition between radio-labeled antigens and unlabeled antigens for binding with a limited amount of specific antibody. In figure 1, the concentration of unlabeled antigens is higher than the concentration of radio-labeled antigens. As a result, few unlabeled antigens will be displaced from the binding sites on antibody and the exposed sites will be attached to the radiolabeled antigens. Because of this, following incubation, after removal of the unbound antigens, the radioactivity of the bound antibody will remain low. This is because few radiolabeled antigens are attached to the antibodies. By contrast, in figure 2, the concentration of radiolabeled antigens is higher than that of unlabeled antigens. As a result, more unlabeled antigens will be displaced and more radiolabeled antigens will be attached to the antibodies. Because of this, following incubation after removal of the unbound antigens, the radioactivity of the bound antibodies remain high. This is because more radiolabeled antigens are attached to the antibodies. For example, we want to estimate serum insulin. For this, we have to extract serum. Insulin present in the serum is the unlabeled antigen. Unlabeled means the antigen is not radiolabeled. In addition, we also require complementary antibody and radiolabeled insulin radiolabeled insulin. Radiolabeled insulin is the radiolabeled antigens. First of all, the well of a microtiter plate is coated with antibodies specific for insulin. In next step, the sample serum is added. The serum contains unlabeled antigens. Following this, these unlabeled antigens will bind to antibodies. This is followed by the addition of radiolabeled antigens. As a result, the radiolabeled antigens will compete with unlabeled antigens for binding with antibodies. As a result of this competition, some of the unlabeled antigens will be displaced and the exposed binding sites will be occupied by the radiolabeled antigens. After incubation for a predetermined period, the unbound antigens are washed off and then the radioactivity of the bound antibody is measured. This value is then compared with a standard calibration curve in order to estimate the concentration of insulin in patient serum. Now the question is how to prepare a standard calibration curve. For this, increasing known amounts of unlabeled antigens are added to a series of test tubes containing the same amount of radiolabeled antigens and complementary antibodies. Now the unlabeled antigens will compete with radiolabeled antigens for binding sites. As a result of this competition, some of the radiolabeled antigens that are attached to antibodies will be displaced and the exposed binding sites will now be occupied by the unlabeled antigens. The relative amount of radiolabeled and unlabeled antigens that are attached to antibody is a function of the relative amount in the series of test tubes. After incubation for a predetermined period, the unbound antigens are washed off and then the radioactivity of the bound antibody in all those test tubes is measured by a gamma counter. After this, a standard calibration curve is plotted by taking the concentrations of unlabeled antigens on x-axis and percent antibody bound to labeled antigen on y-axis. From this calibration curve, the concentration of insulin present in patient serum can be easily determined. Friends, this is all about the principal procedure and applications of radioimmunoassay. For more updates, please subscribe my YouTube channel, Sam Biology Class.